All right, let's dive right in. Today we're taking an architectural tour of the cell. Think of this as a visual study guide where we're going to treat this fundamental building block of life like a microscopic city to really understand how it's all put together. First things first, you've got to stop thinking of a cell as just some simple blob of jelly. No way. Instead, I want you to picture a bustling, microscopic metropolis. It's got everything: a government, power plants, factories, even its own border patrol. Every single structure has a precise job, and it's this intricate organization that makes the cell the fundamental unit of all life.
So, here's our tour itinerary for today. We'll start with the city's basic infrastructure, then head straight to Command Central, that's the nucleus. After that, we'll explore the industrial zone, check on the power and waste utilities, and finish up by comparing the city's key defensive walls.
Okay, first up, every city needs a solid framework and a clear border to even exist. Well, in the cell, that's the job of the cytoskeleton and the cell membrane.
The cytoskeleton is pretty much exactly what it sounds like. It's the cell's skeleton. Imagine this incredible 3D network of protein fibers acting as internal scaffolding, giving the cell its shape. It's built from three main types of fibers. You've got thick microtubules that act like highways for transport, thin microfilaments that help the cell move, and super tough intermediate filaments that provide strength. This network isn't just for support. It actually anchors all the other organelles in place, so they're not just, floating around randomly. Next up is the city's border patrol, the cell membrane. Now look at this. It's not some rigid brick wall. It's actually a flexible, fluid barrier made of something called a phospholipid bilayer. This structure defines the cell's absolute edge, separating the internal environment from the outside world and acting as the ultimate gatekeeper for everything that comes in and out.
And here's where it gets really cool, how these two work together. You have this flexible, almost fluid-like membrane on the outside, and this supportive cytoskeleton on the inside. That combination is absolutely key, giving the cell this fantastic balance of stability and elasticity. It means it can withstand physical stress without just falling apart. All right, with the city's framework in place, our next stop is its most important building. Welcome to Command Central, the nucleus. This place is basically City Hall, the main library, and the CEO's office all rolled into one. The nucleus is where the cell keeps all its genetic material, its DNA, organized as chromatin. Its primary job is to protect this precious blueprint and control the cell's activities by regulating which genes get turned on and off. Protecting that vital DNA is a high-security barrier called the nuclear envelope. Now, unlike the cell membrane, this is a double membrane, 2 layers. This separation is a massive evolutionary advantage. It lets complex cells physically separate where the genetic code is read, that's transcription, from where the proteins are actually built, that's translation. This allows for so much more control. And for extra strength, it's supported on the inside by a protein mesh called the nuclear lamina. But how does anything get through that high-security wall? Well, through these gateways. And these aren't just simple holes. They are massive, incredibly intricate structures called nuclear pore complexes. Think of them as the highly regulated security checkpoints controlling all traffic between the nucleus and the cytoplasm. And I mean it. These pores are the only way in or out. They selectively transport large molecules like RNA and proteins, making sure that only the right signals get in and the right instructions get out. This regulation is absolutely critical for controlling which genes are turned on or off at any given moment. Okay, now that we've seen Command Central, let's head over to the industrial heart of the city, where all the manufacturing and shipping takes place. This is the endoplasmic reticulum, or just the ER for short. It's the city's main factory complex, and it comes in 2 distinct types. First, you've got the rough ER, which is usually located near the nucleus. It's called rough because it's studded with these little things called ribosomes, making it the protein factory. In contrast, the smooth ER doesn't have ribosomes, which gives it that smooth, tubular look. It's the city's lipid factory, but also a crucial hub for detoxification and storing calcium ions. So once products are made in the ER factories, they need to be processed and shipped. That's the job of the Golgi apparatus. Seriously, just think of it as the city's central post office. It's a stack of flattened, membranous sacs called cisternae, and it receives, modifies, sorts, and packages proteins and lipids for delivery. So here's how the assembly line actually works. Products made in the ER get loaded into these little transport bubbles called vesicles. These vesicles travel to the Golgi and fuse with the receiving side, which we call the cis phase.
As the cargo moves through the Golgi stacks toward the shipping side, or the transface, it gets modified and sorted. Finally, new vesicles literally pinch off from the transface, all packed and ready for their final delivery. You know, every city needs its utilities, so let's look at the power plants that keep everything running and the sanitation department that keeps things clean. And here they are, the famous powerhouses of the cell, the mitochondria. These are the city's power plants, plain and simple. Through a process called cellular respiration, these double-membraned organelles generate the vast majority of the cell's energy currency, ATP. This is the fuel that powers pretty much every single activity in our cellular city. And here we have the sanitation crew, the lysosomes. These are essentially little bags of powerful digestive enzymes. They act as the cell's garbage disposals and recycling centers, breaking down worn-out organelles, food particles, and, crucially, defending the cell by destroying invading viruses and bacteria. Let's actually watch these lysosomes in action during a process called phagocytosis. So a white blood cell engulfs a harmful bacterium, trapping it inside a vesicle. Then, a lysosome comes along and fuses with that vesicle, releasing its powerful enzymes to completely dismantle the pathogen. It's a perfect example of cellular defense at its finest. For our final stop, let's synthesize what we've learned by directly comparing the two most important protective barriers in our city, the outer wall and that high-security inner sanctum.
So let's really break down the key differences here. The cell membrane is a single bilayer surrounding the entire cell, regulating all traffic. In contrast, the nuclear envelope is a much more robust double bilayer that only surrounds the nucleus. Its specific job is protecting the genome. You see, one guards the entire city from the outside world. The other guards the crown jewels within the palace. Both are essential gatekeepers, but for very different reasons.
And that brings our tour to a close. This quote really says it all. The intricate, organized architecture we've just explored, from the outer membrane all the way to the inner nucleus, is what makes the cell the fundamental unit of life. Without this incredible structure, there is no function. I'll leave you with a final thought. We've seen how precisely engineered this cellular city is. But what happens when the genetic blueprint for just one tiny component is flawed? A single faulty protein in a nuclear pore, a missing enzyme in a lysosome? This is the very basis of many genetic diseases, a powerful reminder of how critical every single architectural detail really is.